All right, great. Welcome back. Very happy to have Sally here. Sally is also from UBC. She's a professor, very well known, very well accomplished, etc., etc. You can read about it online. She's a Wikipedia page, which is very good. But we wanted Sally for a long time for this course, and we're very happy that we finally managed to get her here. Enjoy her lectures, enjoy talking to her. Sally.
Thank you. It's been really wonderful chatting with the students that I've chatted with so far, and I'm looking forward to chatting with you all. So I'm going to go back, and some of it will be a little bit of review, but then I want to get us really strongly grounded in evolution by natural selection. But first off, what's evolution? And both Kavita and Deepa touched on this yesterday. Evolution is change, just change in the form of organisms, behaviors, phenotypes over time in a population. So that change is what Darwin called descent with modification. So it's really the core of evolution is just about change. It's not by natural selection necessarily. A lot of times people equate selection with evolution, but they're not equivalent. So evolutionary change can occur for any number of reasons, and you guys covered this yesterday. It can be chance bursts of reproduction, what Kavita was talking about, drift, sampling by chance. It can be mutations, it can be migration. But to understand how organisms survive and reproduce, how they match the environments in which they are found, none of those other processes can do it. The only process in evolutionary biology that can come to explain the match between organisms and the environments that they live in is natural selection. So natural selection is a process whereby some individuals contribute more offspring to the next generation as a consequence of their carrying a trait or traits favorable to survival or reproduction. So when you see these amazing matches in the world between organisms that are fit to the environments in which they live, I look at that as an evolutionary biologist and go, wow, that's reflecting a long evolutionary history where mutations arose. Some of them would have been lost by chance, as Kavita mentioned. But those that did rise to fixation are preferentially the ones that improved survival and reproduction in that environment. So this subset of evolution, evolution by natural selection, requires three things. And these three principles can underlie this lecture. The first is that individuals vary in some trait. If there's no variation, selection will still act, but it's acting on nothing. Everybody's the same. Everybody has the same chance of surviving and reproducing. It's not that selection's gone, but you're not going to have evolution by natural selection. You're not going to have change. Individuals with some trait values are more likely to live and or reproduce. Without that, there's no selection. If everybody's equally fit, then it's chance, it's drift, that like Kavita was talking about. And then finally, parents have to have offspring with similar trait values. You have to have heritability. And if you have these three premises, if they're acting all together, then by necessity, you're going to have the individuals that survive and reproduce are going to be a subset of the parents. They're going to have offspring that are more similar to them, and the next generation is going to be different. So you will have evolution by natural selection when all three of these are met. So I'm going to use a case study today. I've been working with a team on COVID modeling over the course of the pandemic, and I'm going to talk about estimating selection for this on this virus. And so this is going to be my case study for the lecture. And pretty much, within a month, we had the sequence of this viral genome immediately after these first reports of an unusual respiratory illness in Wuhan, China. And immediately when that sequence was produced, scientists were looking inside for signs of selection. How has this virus been selected? How is it different? How is it different from the types of coronaviruses we see in bats, pangolins, and other species? And today we have more information about this virus and its evolution, including by natural selection and drift, than we've ever had before. So this is there's over 16 million genomes in this GASAID. I'll refer to it often as the genomic database for a bunch of infectious diseases. That's the ID. But for the coronavirus, genome database has been going onto GASAID. So never before we had such a full picture of evolutionary change. And just to ground us on, well, what kinds of changes are we seeing? I'm going to show you some pictures that are drawing the phylogeny. This is kind of a funny phylogeny, so let me orient you. Phylogenies are evolutionary trees relating sequences to closely related sequences. And so the branches, each dot is a virus sampled from a single individual. The ones that are closely related, that the sequences are very similar, then you draw lines that go not so deeply back. The ones that are more different, you have to trace back further until you get to their common ancestor. We can talk about that another time. So this tree is radially symmetric, where each of these circles represents time. And the samples were sampled from the beginning out through the first year. And what you can see when they drew these phylogenies is that actually the common ancestor of all the samples came from late 2019. So it wasn't like it was a disease that was circulating in the world for many months before that we didn't know about. We would expect the most recent common ancestor to be older, and it wasn't. It was pretty much where we expected it to be. The other thing you can do is we know how time is passing, and not shown here, but we also know how many mutations are accumulating over time. And so we can just count that and estimate the mutation rate per unit time for this virus. Roughly once, somewhere in the genome every 2 weeks, it mutates. As an evolutionary biologist, when I look at this phylogeny, it's really star-shaped. That's the technical term for what you see when in a growing population, pretty much all lineages are equally likely to be successful. Everybody seems to have good representation in this star-like phylogeny. That's not what you see with selection. With selection, some lineages do much better than others, and you don't see that kind of symmetry. But in the first year, there weren't really strong signs of selection. It seemed like almost every one of these viruses, even though they were genetically distinct, was surviving and reproducing equally well. And then along came alpha. Let's see if this will run on its own, or I have to press something. There we go. And so on December 21st, 2020, a year into the pandemic, for the first time, we had really strong evidence of selection acting on this virus. And by strong evidence, what I mean is that the same rise in frequency was observed week after week, and in health district after health district here shown in England. So this particular coloration is a frequency of a particular marker, and this is just showing that you're seeing the same frequency rise over space and over time. Drift can lead to rises in frequency. It often does. Half the time, alleles go up, and half the time alleles go down. So a rise in frequency isn't enough to show that it's selection. It could have been a super spreader event, any stochastic thing. And so to be able to distinguish selection causing a rise from drift causing a rise, you really want to see evidence of it again and again, time after time, week, place after place. So here, this was a first variant of concern, and it was the strongest demonstration of selection. The second year of this pandemic, the tree has entirely changed. With the rise of alpha and then it spread, you can't even see that in this tree because it's then got dwarfed by the rise and spread of delta, which then has gotten dwarfed in the third year, late in the second year, and in the third year by the rise and spread of these Omicron lineages. So this is what you see in a phylogeny with selection, where one type is really coming to dominate and pushing out all of the other lineages. So let's go back to those three premises of evolution by natural selection. How do they apply in this case? The variance, well, the variance is pretty obvious. It's caused by the mutations in the RNA sequence of the virus. So they're introduced by mutations. Selection, we can estimate how much more transmissible the alpha variant was relative to the previous Wuhan strain. And estimates are somewhere in the 40 to 80% higher rate of transmission over time, more likely to pass from person to person. And the heritability here is an alpha virus gives rise to an alpha virus. It's encoded in the RNA, so the heritability is there too. So I'm going to mainly talk about estimating selection, but for right now, I'm going to start with a model that I did with Troy Day about predicting what kinds of selection we think are acting on this virus. And so this was early on in the pandemic when we wrote this paper, and we were trying to predict what could happen and really talk about evolution of viruses, because the public discourse, the scientific discourse really was black and white. On the one hand, people were saying, don't worry about this virus. We know all diseases evolve to be milder and milder so they don't kill their host. It'll be fine. Evolution will take us down that route. Other people were saying, as soon as this adapts to humans, it's going to be like Godzilla and wipe us out. And we needed kind of more of a nuanced understanding of what might happen, we thought. So I'm not going to go into too much detail. I might cover this a little bit more on Thursday. But we used a model that's like a classic compartment model called the SIR model, where you have susceptibles, infected, and recovered individuals. But for COVID, we wanted to have it a little bit more realistic. There's exposed individuals before there's virus, enough virus to transmit. So there's another class. And there was a lot of transmission from individuals that didn't have any symptoms. Either they were pre-symptomatic, which meant that they were about to become symptomatic, and some individuals were asymptomatic and just remained unaware that they had COVID. And this was important to include because some of our models were trying to predict, like, what happens if we recommend these policies, like you have to stay at home if you're sick, or you have to you do symptoms testing when you enter into a facility. Well, that will only work if most of the transmissions are happening from this class. So anyway, model. And these arrows represent flow rates. Like this arrow would be recovering from symptomatic disease. This arrow would be death. These arrows represent transmission. They're dotted because it doesn't convert an infected individual into a susceptible or an exposed, but it influences that rate. To go from susceptible to exposed, you have to have contacted individuals with the disease. And so that's what those rates represent. Yeah, so the susceptible has no virus in it. The exposed individuals have the virus, but the viral load is too low to actually transmit to the next person. And so that's like 2 days, 3 days with COVID. And it mainly just makes a slight delay in the dynamics, but is just to make it a little more realistic. Right, so the exposed how do you estimate that? It comes from contact tracing information. Like, when did you actually come into contact with somebody that was likely infected? Because you might not even test positive. You don't have symptoms and the viral load is too low. So that's really where it's coming from, is how, and a lot of that is done within families, within households. You know, when the first person came in with the virus, and then you estimate it. Not something I did. But yeah, that's how contact tracing is how you do it. One of the ways. Okay, I'm going to skip over this because this isn't where I want to focus, but I just want to tell you that you can translate this diagram into math equations. So these math equations are saying, well, let's go to this one. If you're infected, you can move out of the infected class over time. If you die, or if you recover, you'll move out of that class. But you can also enter that class by getting symptoms if you were in the pre-symptomatic class. So these are just bookkeeping of all of those arrows to get us at how every one of those compartments change over time. Just a model. And so this is the black box that I want to use right now, and we can come back to it. Even though that looks like a complex set, we can actually estimate selection for a new mutation that does something. So a new mutation can make it more transmissible, Or maybe it expands how amount of time it takes in the exposed class or makes it shorter. And we can say a mutation has some arbitrary effect on this picture. How strongly is it selected? So that black box I might touch upon on Thursday, but for now I just want to say we get an equation for the selection over time, and we can understand the elements of this formula. In particular, these things with a delta is what the mutation does. Does it increase transmission? If so, that delta beta is higher. Does it increase the recovery rate? Then the delta kappas are higher. Anyway, we can summarize all of the effects on this life history.
The number of recovered also influence the numbers.
Yeah, and this, I should have emphasized this. This model was during that initial phase when there were almost everybody was susceptible, and there were very, very few recovered individuals. Over time, you have to deal with waning of immunity and those recovered individuals looping back to the susceptibles. But this was during that first year, before vaccines were introduced, and we didn't have to deal with that.
Parameters in this.
Yeah, tons, yeah.
Okay.
Oh, I haven't even counted them, but all of those arrows, yeah. Yeah.
They are like,
So there was data on a lot of those parameters, and then there was like one core one we couldn't estimate, which was the transmission rate. And so there was one scalar for that that we could figure out by how fast the virus was spreading. But let me tell you what the take-home message was, because we can look at, we looked at this equation, and we're able to say, well, what if it increases transmission? What if it does this? What if it does that? And I'm just going to summarize what the take-home points were, and we can talk about that maybe more on Thursday. So selection on this virus was positive if the virus mutated and increased transmission. That makes sense. If it's able to get from person to person at a higher rate, then that's going to be positively selected. And we were able to show that, yes, anything that increases those betas will spread due to selection. The other thing that is favored is increasing the length of these stages. The longer the individuals hold the virus, the more strongly favored they are too. And so we were able to measure all that. But I think the key thing that was a take home at the point was actually selection on this virus to become more deadly was almost gone, was almost zero. It was not selected to increase virulence. Virulence is your chance of dying from the disease, nor was it strongly selected to decrease virulence. It was almost zero. So why was there no selection on this virus to become milder or Godzilla? And the reason was for the parameters of this model, the virus, what we know about COVID was the virus load was rising and falling before the severe illness, critical illness, and death. And so by the time that virus was killing us, the virus was long gone. There was no evolutionary future left for that virus in those hosts. It was past that peak load of virus transmission. So the evolutionary selection is focused on the majority of when the virus is actually present and replicating in the body. Once the immune system kicks in and the virus load has gone down, you can still have symptoms, and you can still have side consequences, but it's not going to, the evolutionary future, the evolutionary selection pressure acting on the virus isn't responding to whether it kills us or not. It's a side consequence as far as the evolution of the virus is concerned. Yeah. No, the mortality was due often to an immune overreaction or damage to our lungs. And most of the damage to our lungs was due to that immune cytokine storms and other immune responses. Yeah, but that was after the immune system successfully cleared the virus. Yeah. But that doesn't mean that the virulence couldn't evolve. It just wasn't being directly selected to go up or down. Instead, we argued that it would probably evolve up or down by pleiotropy, by mutations that increased transmission rate, and as a side consequence, they affected mortality. And so we predicted that there are two ways that that pleiotropy could act. It could be that mutations have a higher transmission rate because the virus replicates better. It gets into our cells better, replicates better. But then as a consequence, we would expect positive pleiotropy. The virus does better in our bodies, is more likely to rise to high numbers and kill us. If it transmits better, gets into our cells better. But there's an alternative. And at the time that we were writing, COVID was largely a lower lung infection. That was causing the oxygen deprivation, and the severe attacks was the deep lung infections of COVID. So there was another possibility that actually it's not that easy for a virus to transmit from deep in our lungs. It's not very near our nasal passages. If it evolved to better attack cells in our nasal passage, which have different types of ACE2 receptors, then maybe it could transmit better, but be less virulent if it evolved a change in tropism, where it acted from deep lungs to nasal passages. And that would be a negative pleiotropy. The virus is selected to transmit better, but mutations that cause it to switch where it normally infects have this, as a side consequence, a lower virulence. And since that time, we've had more and more, unfortunately, variants of concern, and we've seen both. So I think this evolutionary model was insightful because it scoped out what the realm of possibilities are. For alpha, beta, gamma, and delta, they do attack our cells more readily. So there's a higher transmission rate, but they're also more deadly, both higher hospitalization rates and death rates. And these are among unvaccinated, not recovered individuals, individuals that have not had either the disease or the virus or the vaccine. But then Omicron hit, and that changed tropism from lower lungs to the nasal passages, and that decreased both hospitalization rates and death rates, even without a vaccine. And it's not zero, right? You don't want even half the death rate isn't great relative to the Wuhan strain, but it is lower. And all of the strains that we've seen since then are still in the Omicron family. Delta's not extinct. We still kind of every once in a while see it pop up, but by and large, everything is Omicron now. Okay. So that was some predictions from some evolutionary models. And it's a little bit of a sophisticated model, so I went kind of hardcore on you at first, but I just wanted to see how you can build models and estimate what selection is predicted to be. Now I want to switch to, okay, what is selection on these variants? And so if you go onto GASAID on any particular day, you can download sequences and estimate the number of sequences on any day that are of one type or another. So this is an old graph, but BA2 was pretty predominant in the spring of 2022. So those circles are bigger. There was more samples. So this is on a particular day, like April 1st, how many samples collected on that day were BA5 versus BA2? And then on the next day, on the next day, on the next day, and so on. And so you see these BA2 is falling. You think, okay, that's due to selection. It doesn't look like drift. Doesn't look like those wobbly graphs that Kavita was showing. What is the selection coefficient? So if you know only one equation from evolutionary biology, I would argue this is the one to memorize. And it's basically in a formula form, those three premises. What this is that the rate of change of the allele frequency. So dPdD is 0 if there's no change in the allele frequency. It'll be positive if P is going up. It'll be negative, the slope over time if P is declining. P being the allele frequency, let's say here, of the BA5 type. And I'm going to, because these are asexual viruses largely, I'm just going to talk about the lineage. So not a particular mutation, but just ask how much selection is acting on the lineage. Okay, so this formula, dPdT, SP times 1 minus P, or SPQ, This is evolutionary change by natural selection. I said that there had to be variation. P times 1 minus P represents variation. If P is 0 or P is 1, no variation. And Kavita mentioned the binomial. If you drew one allele by chance from a binomial distribution, then the variance that you would expect with n equals 1 is P times Q from the variance of a binomial. And throughout, I'll be using 1 minus P to stand for Q, the frequency of the alternate. So that's the variance, and this is the selection coefficient. And it can be solved, according to this formula. And I'm going to come back to these slides and say, what was the answer? What was the selection coefficient? But I want to stop there, because how many people feel like they could derive these equations? Good. Because I want to get you comfortable saying, no, actually, I'm comfortable with this. I can derive it. This. All right. I'm going to start somewhere funny. I'm going to start not with evolution. I'm going to start with ecology. And then the reason for that is that I think more people are comfortable with exponential growth. How many people are comfortable with exponential growth of a bank account or a population? A little bit more comfortable. Okay. So let's start with population growth. We have some number of individuals. So this is the number of individuals. And then rate of growth of that number in the exponential growth model is just R times N. This is like the rate of growth of your bank account, depends on the interest rate your bank is willing to give you, times how many dollars you have or rupees you have in your bank account. And if R is positive, it's going to grow. You don't want a bank that's going to give you R negative interest. Don't go there. So this equation, I'm not going to do many solutions to these kind of equations, but this is a differential equation because it depends, it says what the variable, how the variable changes with respect to its current state. But I'm going to teach you one trick for solving this equation, and it actually works for solving the other one I'm going to show you later. So normally, dn dt represents a slope, a rate of change per unit time. But just pretend for right now that it's a fraction. So if it were a fraction, then we could say, well, if it were a fraction, I can move everything that involves n to one side and everything that involves t to the other side. I'm just moving this over there and that over there. And I've actually separated the variables. N's on one side, t's on the other. So one trick for solving this type of differential equation is that you then integrate this. But you integrate them with respect to the variables that are now separated. The integral, this may be, for those of you who are comfortable with calculus, this may be familiar. This would be log of n. And I'm going to write ln for natural log of n. The integral of r with respect to t would be r times t. And there's a constant of integration. I think I put on my Slack channel some refreshers of calculus if you have had it, and then it goes through all the rules of calculus if you haven't had it. Anyway, rule of calculus. So when you, at this stage, you take the you exponentiate both sides. E ln of n is e to the rt plus c. And the reason I exponentiate it is exponentiation undoes the natural log. And this just gives us n on this side. So I wanted to do the antithesis to get rid of it. And on this side, there's another handy appendix has rules of algebra. And one of the rules of algebra is that you can expand exponential terms that are e to the a plus b as e to the a and e to the b. But what's this pesky constant of integration? I don't want, that doesn't make any sense for a biologist. What is this C? Well, we normally solve for those constant of integration by saying, I know something at some point in time. And so, for example, what's typical is you say, at t equals 0, I have some initial population size. So here, if t equals 0, I get n of 0 is equal to e to the r times 0, e to the c. And e to the 0 is just 1. So this goes to 1, and that lets me know what EC is. E to the c is just n naught. And that, in the end of the day, and I'm going to write it up here because we're going to use it later, is that if we have an equation dn dt equals Rn, that we can solve it as n at any future time t is equal to e to the RT times it's wherever we start. And here I used a subscript to determine specified time, and here I'm being more explicit about when time. Yellow chalk. Okay, I'll throw this away. Okay. Okay, comfortable? Questions? So this is why it's called exponential growth, is this formula. E to the RT will grow exponentially. It'll look like this if R is positive. It'll look like this if R is negative. And it'll look like that if R equals 0, staying constant wherever I start. So that's why it's called exponential growth. Questions? How am I going to relate this to evolution? Imagine now there's not one type, there's two types. And you have, let's say, a vat of cells inside a beaker, and it consists of two types, each of which is at low numbers and growing exponentially. What would happen over time to the frequency of one type versus the other? And that's what I'm trying to do with the COVID models, is estimate how one type is growing faster than the other type, what's selection acting on those two types. So I'm going to say now, same sort of results. I'm just going to apply that logic and say, what if P at time t is equal to the number of cells in my vial that are of type 1 or type A over the sum total number of cells. Maybe I'll use big A and little a just to be consistent with. So that's just counting up the number of cells in my flask at any point in time. But we've just solved for what n is at time t. And so we can simply plug in the exponential growth of the first type, which happens at rate Ra times t, and the initial number of cells that we started with of type A. And then on the denominator, we have the same thing. This is the prediction over time, starting with an big A of the A type and a different type of cell or a virus or anything that is of the little a. And this will work for a haploid model or for an asexual virus. But I'm not dealing with sex, I'm not dealing with recombination, I'm not dealing with two loci. I'm dealing with a model where the types breed true. So when I count big A and I say that it's growing exponentially, all of those kids are big A. So I've got to be clear about that. So this is what I call a mixed up equation, because this is on the allele frequency side, and this over here is on the number side. But we can divide the top. You're always allowed to multiply or divide top and bottom by something. It's just multiplication by 1, and we're always allowed to do that. So I'm going to divide the top by the sum of the numbers initially. And I'm going to divide the bottom by that same number. So that's just multiplication by 1, and I haven't changed anything, okay? But what is NA0 over the sum of the number of cells at the beginning in terms of P? This part here. P at time 0. Good. And on the bottom, this is all the same thing, P at time 0. And what's this one? N little a divided by the sum. 1 minus P0. Or Q0, doesn't matter which way you want to write it. Okay. Oh. But does that make it harder for you all to see? It's okay? All right, so I'm going to write this over here because it's another one of our important equations that I'm going to want to keep. And that is, we've now said that P at time t is equal to e to the r. Oh, whoa, whoa, whoa. I can simplify this first. I want to simplify it first. So this right now is relating evolutionary change to the growth rates of the two types. But imagine dividing, so now that we've got this equation, we've replaced it for the P's and Q's. But I can divide, I have two R's. And what I want to show you is I only need one measure of the relative growth of the two. So I'm going to divide the top and the bottom, let's say by the growth rate, the growth over time of the little a allele. Then that will lead to e to the r a over t, e to the r little a over t times p naught. And this thing here, using the rules of exponentiation, can be simplified as e to the rt minus r little a to the t. Just because whenever you multiply two exponential functions, you sum their power terms, and when you divide, you subtract the power terms. And this is just, can be factored as so. So on the top and on the bottom, that's all that matters. Not the growth rates of the two populations themselves, but the difference in the growth rates. So I'm going to call this S for selection. The difference in those exponential growth rates is a measure of how much selection is favoring the rise in one type over the other type. If they rise at the same frequency, then S will be 0. Those R's are the same. So let me write, that's the form I want over here, and I'm going to write e to the st p naught over e to the st p naught plus 1 minus p naught. All right. Let me get some erasing done. You can think about that and ask me questions while I'm erasing. So the key point is that if the populations were growing exponentially, it doesn't matter if the whole population's growing or the whole population's shrinking or the whole population staying constant. Evolutionary change is predicted to occur according to this formula that only depends on the difference in those growth rates, where S is R big A minus R little a in those exponential growth rates. What makes you uncomfortable about this model? I've got different directions I want to go next, and now your discomfort can help me move. Yeah. Yeah. Very good. So everything I'm going to talk about is the very large population deterministic dynamics. So you can think of that as the mean of the dynamics that Kavita was going to talk about. But yes, when a mutation appears, it appears in a single copy. Yeah. Yeah, very good. So if an allele arises, even in an infinite population, if a mutation arises in a single individual, there's still a finite number of them. And so you typically have these periods of time where maybe it was lost, and it takes a while for the mutation to arise again, and maybe it's lost again. But on the third try, maybe it does rise. And then following its rise, those in a large population, those dynamics smooth out. And how much wobble you see depends on how large that population size is. So I'm really focusing on this phase, not the phase when the numbers are small, because then you need to deal with the probability that you don't lose the allele when it first appears. Absolutely. That's what you showed this morning with Kavita, is that even if S is positive and we expect it to grow, it won't necessarily grow because it's lost right away. So typically what we do is we say there's this danger zone when there are very few copies, and we have to deal with that stochastically. But then once there are a lot of copies and the population size is large, then we go to these equations. Next thing you're a little uncomfortable with. What was that? You didn't like exponential growth. Good, you get to. How many populations can't grow exponentially forever? And if all of evolution rested on the assumption of exponential growth, we would be in big trouble. So what happens if there isn't exponential growth? So I gotta, all right, I'm gonna do this a slightly different way. So we've got, well, I want to generalize this now, where we have two types, N1 and N2. And they're not growing exponentially. Let's say that they grow exponentially when the population size is small, but there's some function of the number of cells in my vial that causes the growth rate to decline over time, or as the population size grows. So this could be, if I was doing the logistic model, that would be minus R n over K, but it could be any other model. So I'm just going to keep this general right now and say, I'm not even going to say what's causing the departure from exponential growth. It could be competition, could be toxic interactions, but they have some sort of form that depends on the number of the two types. And here's the core thing that I have to do for my second population. If both types, their growth rates go up or down with the population size in the same way, then I'm going to show you that you get this equation back. But if they don't, like maybe one type is a better competitor and the other one's a terrible competitor, those aren't going to be the same functions. Then we can't separate the ecological dynamics from the evolution. We have to do both. And there are fields of exploration looking at evolutionary ecology, doing the two of them in a lockstep. But I think it's useful to realize that if this assumption is true, that yes, there can be competition, but it's affecting their growth equally, then selection and the evolutionary equation remains unchanged. So this is going to be a proof of that claim. You don't, I'm just going to show you the nature of the proof, not just so that you can not see that it's a mystery. So what I want to do is I want to get here. Hold on a second. I want to see if there's an intermediate step that will be faster. Yeah, there's going to be an intermediate step that's going to be faster, but let's get there. Okay. So dp dt, I don't know what p is. If I knew what p is, I could take the derivative of it. I don't know what p is. That's the whole goal. But I do know, by definition, it's the sum of the two types. And my notation is bad because I'm flipping back and forth between whether I'm referring to these as alleles or just ones and twos. In your own notes, do whatever is comfortable. How many people remember or ever were taught the quotient rule? How do you take the derivative of a fraction? So this is a quotient is a fraction, and if you represent it as I'm going to say g over h because sorry. If this is a fraction, then we know that the derivative of that fraction with respect to our independent variable is equal to, you take the derivative of the numerator times the denominator minus the derivative of the denominator times the numerator, and divide everything by the denominator squared. Just a rule of calculus. That's how we can go from fractions, take the derivative of it. Okay, so, but I know, okay, so we're going to apply that in this case, and that's going to say dn big A dt times the denominator minus the derivative of the denominator. The derivative of a sum is the sum of the derivatives. So I'm going to use a calculus rule there, but the derivative of that plus that is the sum of the derivative of the first one plus the derivative of the second one. So that gives us a derivative of the denominator, that part, but we need to multiply it by the numerator. And then that's all over n big A plus n little a squared. Okay, so a cleanup. I always like to scan and see if there's anything that's kind of in common. And if you notice, there's this same derivative is here and here, and it's multiplied by n big A in both cases. So just scanning says, oh, I can simplify this before getting any further by canceling that term with this term. Just to make it a little bit cleaner. But these derivatives, well, let's keep them that way. So that's d n big A dt times times n little a plus the derivative of the other thing, dt times n big A. And then that's all divided by their sum squared. Not too much longer on this proof. But we know what the derivatives are. And we know them in general, even with how each one changes, even if there's density dependence or competition going on. So I'm going to plug those formula in now. So we have R big A minus this function. It's a function of the population sizes. It's not necessarily a function of the total. It might depend more on one or the other. One might be a bigger cell, one might, doesn't matter. It's the same function for both types of cells. So I'm just going to write it as that. And then that all is multiplied by n big A. That's this term here, plug in, gets multiplied by n little a there. And then same thing for the other function. It is our little a minus that density-dependent function, whatever it was, and then times n little a from over there. So this part is this. This part is that. And that gets multiplied by n big A. Thank you. And that's here too. And that minus sign is in the quotient rule. Thank you. Because the next thing we're going to want to do is scan, and this is all divided by the sum of the squared. Scan it. Anything else that you can see would go away? Okay. Get rid of the f. It's minus f times the product of the numbers. Minus minus f is a plus f times the product of the numbers. So the density dependence doesn't matter. We didn't even specify what kind of density dependence we had. The only thing we had to assume was the growth rates were equally affected by that competition. And then how else can I simplify this? Yeah, so go ahead. And let's simplify it one last step. Yeah. In that notation, or if we're just calling P the frequency. Yeah, excellent, excellent. So here is this fundamental formula for evolutionary change, dP dt is SPQ, SP times 1 minus P. We derived it initially, we were thinking about it from exponential growth model. So if I set the F's to 0 and didn't have them and carried out these calculations, I'd still get that formula out, right? So this formula would apply if there's exponential growth. But what we've just shown is it doesn't require that. It just requires that the population's doing whatever it does and affecting the growth of both equally. Okay. And a different way, we already showed that the exponential growth model is solved by this equation. So this has this solution for exponential growth. And now we know that it's also got this solution, even if it has density dependence, because this doesn't depend on F. And there's different ways to do it. Remember when I said there was a separation of variables? If you wanted practice and you're good at calculus, then use separation of variables. And that's another way you can prove that the solution to this equation is that one. But I did it a different way that didn't require so much calculus. Thoughts? So let's recap. If two types are growing by whatever means, but their growth rate is different by the difference between their growth rates is S, then whether the population is growing or constant or declining, we're going to see evolutionary change in the composition of the population according to this equation, that the rate of change is going to be SPQ, and over time, we would see this. Okay. So I think that's one kind of interesting way of deriving this fundamental equation for evolutionary change, starting with kind of what you think first principles from how populations will grow. And it helps us connect and know how evolution and ecology interrelate. And I think that's useful because a lot of times students are uncomfortable saying, why did we not pay any attention to whether the population was growing or shrinking? Doesn't that matter? And this is saying, no, actually it doesn't matter as long as our assumption holds of equal effects of density on the two. Anything else troubling you? Yeah. Yeah, so if you have more than 2 types, then what's the easiest way for me to solve this? Let me just note a couple of things, and then this will. So if we had multiple types, let's go to the next. I'm going to do one other thing, and it'll be easier to prove. It's easy to do here, but I have to do more calculus. I'll be able to do it with algebra in the next. So park that. Any other questions? How many people are uncomfortable with derivatives? Oh, one honest person. So there's, but the several of the models that we've talked about aren't, yes. Yeah. Yes. The change in the frequency. So if the populations are growing over time, but one's growing slower than the other, then, but you start at a high number of them. Then initially it might be at a high number, but over time it's becoming a smaller and smaller fraction. So what we're paying attention to is this frequency of all of the types. That's of one type versus the other. Yeah. And so we're really focusing on what's the evolutionary change in the composition of the population. Ecologists care about this, the number of the total population. But right now we're just saying what's happening to the composition within it, the variance within it. The 2 types. Let's do it. So there's 2 very classic models, modeling types of deterministic model. These are differential equations, and they assume there's kind of constant population growth and change. We're treating time as continuous. There's constant births and deaths. And that's pretty good for if you've got bacteria or yeast in a vat. They're constantly reproducing. I want to just show you roughly what happens if the population is instead finite, has discrete generations. Like many animals and plants, it would be annual. We don't want to track them as if they're growing continuously. But I'm going to just do this very briefly, but kind of the idea is similar. And say, what if we have each type reproduces according to some function of the number that we had before. So this is mapping where a population was to where a population goes in the next time step, time t plus 1. Those are called recursion equations, and they don't involve derivatives. They're not an instantaneous growth. They're what happens over a year or a generation. This is called the reproductive number. And if we assume that there's some density dependence, but that it's 1, if the population size are small, there's no density dependence, so that goes to 1, then this reproductive number gives you the number of babies per parent in the next generation when there's not competition. And then similarly, we have a second type. The same function. And you might be a little bit weirded out that I'm now multiplying it by this density-dependent factor rather than before I was subtracting it. But just to comment that oftentimes that reproductive number is written in terms of the growth rates as that. And so if we have this function now, we could also represent it as a function that's changing as another function. Let's call this now G. So these are another functions as an e to another function. And we could represent it that way. And so when we're working with growth rates in the exponent, then that's equivalent to taking the difference. But when we're working with how many kids do you reproduce, then we want to multiply it. Just a detail. And you don't have to do that, but if you do anything else, you're not going to separate the ecology from the evolution. So this is the requirement in order to have evolutionary change just depend on selection and not on the population sizes. Okay, so just quickly, I'm going to just take the ratio of the two. And I'm going to do that so I can answer your question about what if there were more than two types. And so if I do that, I've got R big A, G, and big A over R big A, little a, G, and little a. And so the ratio, the one type relative to the other, you can see immediately that the G's will cancel. And they'll only cancel if I've multiplied those numbers in by the density-dependent factor. So your number of offspring declines by a factor because of the same factor because of density dependence. But then the G's are gone. So immediately we've gone from a general ecological model to one where we only need to know our big A relative to our little a. And so in this we can also say, well, if I could divide the top and bottom by the total numbers, and then that would give me the frequency on top of big A and the frequency of little a on the bottom. Just dividing the top and the bottom by the population size of the two types. So this is the ratio of the allele frequencies, and that's equal to the ratio of the numbers. If I have 10 times more big A's, then the frequency of big A is 10 times more than the frequency of little a. So this is at time t. And this similarly is the frequencies in the previous generation. So notice that, except for here where how I define the two types, if I had 3 or I had 4 or I had 10 times, I could still focus on the number of one type versus the number of the other type, and this calculation would hold true. And so if I'm just focusing in on two types, the relative frequency of one over the other will change over time by that constant. And that's true no matter how many other types there are. Yeah, perfect segue, perfect segue. So here, I was really talking about the absolute fitness, the number of kids this type has and the number of kids this type has. That's really hard to get because you would have to go out there and fully find out whose parents gave rise to which kids and a whole population to know every kid. With plants, you can count seeds, so there's some, it's a lot easier to do like a mark recapture or a subsample to find out how things change from one generation to the next. So it's a lot easier if we had relative fitnesses. But notice that our big A over our little a is the same if I divide all the fitnesses by anything I want. I could divide it by 3.14 if I wanted to. and measure these relative to 3.14. I don't know why I want to do that. Or I could define the fitnesses relative to the growth of the little a type. I can do that. Doesn't matter what we measure fitness is relative to, we will get the same evolutionary equation out. And that's why we say that it's only relative fitness that matters, not absolute fitness. And in particular, if we don't know exactly how many babies a parent had, we typically talk about those as being the fitnesses W big A and W little a, where we're not, those aren't any longer necessarily absolute fitnesses, but they just tell us the relative fitness of one versus the other. Good. All right. So now we have P of T over, for big A, divided by P little a to T is W big A over W little a. Oh, I'm switching time. I'm going to now say at time T, I'm going to switch one time back because I want to now repeat this. So every generation that passes, this is what selection does in this annual plant or annual animal. Every generation that passes, the ratio of the allele frequencies changes by this ratio of the relative fitnesses or the fitnesses. What would happen after 2 generations? If something gets squared, what gets squared? The ratio of the fitnesses, excellent. So I just repeat this, but I know that this at time T minus 1 is the ratio of the fitnesses times what its value was 2 time steps ago. And so now the fitnesses are squared, but I could repeat, and now I can plug in for this, for P at time T minus 1 and keep going until eventually I just get W big A over W little a raised to the power T times whatever their initial frequencies were. And again, I haven't said that there were or were not other types out there. I'm not paying attention. I'm just paying attention to our 2 types. Thoughts about this? So what will happen to this ratio if W big A is greater than W little a as time passes? What will happen to that ratio as T goes really, really high? What was that? Grows and grows and grows and grows. Yeah, so this ratio will go to infinity. Does that mean that the allele frequencies are going to infinity? Yes, the denominator is going to 0, the numerator is going to 1, if those are the only 2 types. Yep. What if W big A is less than W little a? Then this number is less than 1. When we power it up, we'll get 0. When would you expect no evolutionary change by natural selection? Only if they're equal in fitness. Then it stays at whatever initial value it was at. Awesome, awesome. So that is great. And it gets us almost to the end of what I want to do on the chalkboard. There might be one more thing. And that is that this is nice and it gives us the ratio. And then actually, I think it's telling you something fundamental. That the ratio of 2 types grows exponentially. So this is, geometrically in constant time. If we think of this ratio as e to the s, then it would be exponential. So the ratio changes exponentially over time. And that's true in this equation too. If we took p of t and divided it by q of t, you would get e to the st out. But I want to know the allele frequencies. I don't want to know just about the ratio. How would I solve this for p at time t? Now assume there's only two types, so that the denominator is 1 minus p. Let's just write it that way. This is 1 minus p on the bottom. This is raised to the power t and our initial condition. Right. How do I go from a ratio to actually solving for p at time t? I want an equation like this one. Yeah. Yep. P little a is 1 minus p now. And then we've got, good. And now what do I do? Yeah, let's call this for right now, just call it x, so I don't have to write it again and again. So that's x times 1 minus pa. So just expanding, calling that x and then expanding it out. And then you were saying, now you move this term over to the other side. So we get PAT plus x, PAT equals x. Now what do I do? Now I factor out the 1 plus x times PAT and divide both sides by that 1 plus x to get the solution for PAT. Now I have to go back and plug in my x's. So that's W big A over W little a to the tth power times PA. Same thing. And one last step, and we're going to have a nice clean result. I don't like fractions over fractions. It bothers me. So I've got fractions and then divided by fractions. But I can multiply the top and the bottom by W little a to the t times this. And then when I do that, when I multiply top and bottom by W little a to the t, 1 minus p, 0, top and bottom, I get my third fundamental equation that I wanted. At p, at time t, is equal to, this will cancel that and that. So in the denominator, all we have is W. In the numerator, we only have the fitness of a type times its initial frequency. Then on the bottom, when we multiply top and the bottom by this, the 1 gets to be 1 little a, the other type's fitness times its frequency. And then we have to add it to this, but the WAs will cancel and the 1 minus P's will cancel. Okay, we're done. This is a general solution starting instead from exponential growth for an annual plant or an animal that reproduces every year or generation. It will apply as long as there's exponential growth or density-dependent growth that affects both types by the same factor. And you might go, well, won't evolution be entirely different if we're in continuous time or in discrete time? But notice the form of these equations are similar. And indeed, if we said that e to the st is equal to w big A over w little a to the t, they're equivalent. I could just plug that e to the st into there, top, do the same trick with multiplying top and the bottom. I have the exact same equation. And so oftentimes when measuring fitness in discrete time, if we measure it relative to the w little a, we might define w little a to have a fitness of 1 and w big A to have a fitness of 1 plus s. And so in continuous time, then we would replace that with 1 plus s to the power t. And they're not exactly the same. 1 plus s to the power t isn't the same as e to the st, because you get continuous growth, you get cumulative interest growing continuously there. Here you have 1 plus s to the t, but they're very similar as long as s is small. Okay. So those are the fundamental equations of natural selection. The key thing that we've assumed is that a type breeds true. It doesn't have to be a single allele. It could be a whole haploid genome. It could be a virus genome, as long as it replicates entirely to give the same type. There's a lot of math, but I hope it gives you a flavor for where these equations come from and the relationship to ecology. Now that we have them, we can do the fun things that we wanted to at the very beginning, which, yeah. So this was in continuous time, the allele frequency change, and we can then apply that to this data. Because what do I know? I know the data, I know the numbers, and I know of each type, but what I don't know is that S, and I don't know the initial frequency. I know time, I know day, but I don't know the initial frequency, and I don't know S. But on any one day, the probability of seeing the sample I see on that day is given by the binomial distribution with that formula for PT in it. And given how many of one type I see times 1 minus P to the number of the other type I see. How many BA2s and how many BA5s. So we can maximize likelihood for this data to estimate selection. And that allows us to estimate selection for this variant and all sorts of other variants. I just want to show you briefly a website that we keep up and running that does this very thing, estimate selection on the different variants, but we keep it up and running to know how the different variants are rising over time recently. So for example, we see the rise in JN1 towards the present, and its selection coefficient per day is estimated there, shown there as 0.07 relative to those other types. And here's an example where we just generalize it. Rather than 2 types, we actually do have multiple types, but we can, using the same math, just go P1, P2, P3 for all of these variants with their own selection coefficient. You have to specify what your reference is, though. And at this point, I think we're using a particular type that was common previously, HV1, to say that's going to be our fitness of 1. What are we going to measure everything relative to? And this here shows those estimated selection coefficients for different variants that are circulating in Canada over the last 4 months. The amount of data in color is indicated by color. So if there's a lot of data, I'm much more confident in the selection coefficient. Also, the confidence intervals are smaller. If I see if I see evidence of selection in more than one province, we put a little circle on it, which means it's not just spreading in one place, it's spreading in multiple places. So those are the ones with the strongest evidence. And this helps because in Canada, and presumably in India. Over the last 4 months, let's see what the number is, there are 405 different uniquely named lineages. It's beyond alphabet soup. This is too complicated. So we need these tools to go which of these variants is growing faster and which not. And that allows you, like here, they're color-coded, these histograms are color-coded. You can kind of eyeball, clearly JN1 is spreading, that's this blue one. So you can eyeball it, but we knew it was spreading way back here. And we knew what its selection coefficient was, and we could predict when it would come to be more than 50% of the population using this formula. So once we estimated S early on, once we estimated where it started, or when it started, then we can estimate where it was going. And the one last thing I want to show you here is that's going back to the kind of ecology. It also helps us understand so that's the frequencies, how they're changing in relative frequency, but what the politicians want to know, the public health officials, is that going to lead to a new wave? But we can tell them that because even if the population size is declining, we back here, we're saying JN1 is going to lead to a new wave because its selection coefficient is so high. This negative growth rate of the circulating strains, when you add S to it, is going to turn it into a positive growth rate. And we could predict when that new wave would kind of come out from under the other type and spread to high frequency. So we can talk, I'm happy to talk more COVID models with you. I just want to show you briefly that I was downloading from GASE the data for India over the last 4 months. And there's not that much sequence data, but I think it's enough for an exercise. Here's JN1, and this is JN1 and JN1.1. And so I'll put on a little exercise on to ask, is one of those growing faster than the other based on the data that you have? Questions? Yeah. Very good question. It's a kind of surprising how well this worked, right? And I wanted to show you something else. Well, I'm just going to have to use it from here. But it's true with this model as well, that if I paid attention to the ratios, well, I'll just do it quickly. It's so quick. If I had multiple types, but I just am paying attention to 2 types, the frequency of one type at time t is growing exponentially at its selection coefficient. That's its initial frequency. And then it would be divided by the sum of all of the other types. But if I just measure it relative to whatever the reference is, then that will just be e to the 0, because I'm defining selection relative to that reference strain. So that's this formula. This could just be Q if you only wanted to do it for two types. And for Q, there's no e to the st here because we're measuring selection relative to that type. R big A minus R little a, if you remember. So here, the key thing that I wanted to point out about selection, and one of the ways you can tell it's selection, is now imagine in logging this. Logging P times P reference. So this is the target divided by the reference. Then we're going to get SAT. Sorry, then we're going to get the log of this. But this is 1. That's the log of E. That'll undo it. And then when you log E to the SAT times P target over reference, you use the log rules that log of a product is the log of the first term plus the log of the second term. That's the initial time point. The key point is this is a line. Over time, if you take the frequency of something divided by the frequency of a reference and log it, it's called a logit plot, a logit transformation. Selection is super simple. It's a line over time. This is that intercept is telling you that something about the initial frequency. But what that predicts is that you should see selection. Selection should give you over time a line, and you can see it here. This is that logit plot. So this is P, the relative frequency of one divided by the other, and the y-axis is on a log scale. And you can see that it's not bad. These lines aren't bad. That's helpful to know that this distinguished selection from a superspreader. Superspreader events would be flat, and then all of a sudden at one point it would jump up, and it wouldn't fall on a line like that. But you're right, it is remarkable to me that these logit models say that selection is roughly constant over this kind of period, 4 months. And that's only true when we're comparing apples and apples. If a new variant arises, then it's no longer true, right? And because then we've got a new orange that's in the mix. So it's really important to keep track of these variants, and as the new ones arise, measure its selection coefficient separately from the old one. But it is a surprise. And we are at lunch. So I think I have a few more points on selection that I'll just move to the beginning of tomorrow's lecture. But hopefully that gives you a grounding in evolution by natural selection, at least in haploids. Yes, this data, I'll put this one on the Slack. So GASAID requires that there's been a controversy over GASAID, a good topic over lunch. But yeah, you have to get an account for access to that sequence data, but then you can download it. And everybody should be allowed to have access to GASAID. But it's not a public company, and it doesn't have good oversight. And the person that's leading it has been a little bit like, I'm going to remove your access. So some of the really important people had their access removed kind of idiosyncratically. Yeah. Good. Any other questions? Otherwise, grab me over lunch and we can keep going.